Today we're going to be discussing something that we've touched on in several lectures, but we're going to discuss it in detail today, which is the reward circuits of the brain. We're also going to discuss this in the context of drug addiction, which hijacks those same brain centers. Well, we got to define what rewards are, so let's do that. A reward is the motivational property of a stimulus that induces appetitive behavior, which can be defined as behavior related to desire to approach or consume. So appetitive behavior, we're defining it here inside our definition of reward. That's a definition inside a definition. That's two definitions for you. How do you like that? Rewards can be as positive reinforcers. They can be used as positive reinforcers in operant conditioning. And so we've talked about this in context. We haven't talked about the reward circuits yet. So naturally occurring rewards are things that facilitate survival, such as delicious food, like pizza and Reese's peanut butter cups and sushi. Okay. And reproduction, such as sexy time and parental investment. That can be rewarding. I mean, that's kind of the reward of sexy time, I guess. And then that's rewarding in itself. At least that's what they tell me. So intrinsic rewards are unconditioned rewards that motivate behavior because they are naturally pleasurable. It is intrinsic to the reward that it is pleasurable. And then there are extrinsic rewards, which are not inherent and must be learned. So an example of an intrinsic reward is such as delicious food. So some of these naturally occurring rewards. They are naturally pleasurable. They elicit an activity within the reward circuits and make us feel good just because of their very nature. But an example of an extrinsic reward is something that has to be learned. Money is a great example of that. A lot of us like to receive money, but there's nothing intrinsically rewarding about money. We have to learn that money gets us stuff such as delicious food and sexy time, right? So yeah, those are some of the examples of extrinsic rewards. Another extrinsic reward. Some of us feel satisfaction watching the Hokies play on the football field, and when they win, we feel good about that. We didn't do anything for it, and it's certainly there's nothing that benefits us our survival by watching the Hokies play and watching them win. But we've learned over time that we support the Hokies, and when the number of points that they have at the end of 4 quarters is higher than our opponents, hopefully the Cavaliers, then we feel happy. That's another example of an extrinsic reward. There are lots of examples you can think of. You've seen this slide before. So this is the slide where we talked about classical conditioning, Pavlov's dogs. And there are intrinsic and extrinsic rewards here, right? What's the intrinsic reward? Is it the bell or is it the steak? That's right. It's the steak. It's delicious food. So it's intrinsically rewarding to this pup here. Clearly, he likes it. But this pup can learn through reinforcement that a bell can be rewarding in itself. So he's going to hear a bell. It elicits feelings of pleasure and excitement in him because he thinks it means he's going to get food. So now this is an extrinsic reward. So for a long time, there was this hunt to find the pleasure centers of the brain. And the way that this was first really uncovered was in some classic experiments done in the 1950s. They implanted electrodes into a number of brain areas. They figured there has to be some place in the brain that gives us this feeling of satisfaction, this reward feeling. And so they, in order to find it, why not just put electrodes in the brain and then have this electrode connected up to a lever that the rats can then self-stimulate. So every time the rat would press this lever, the electrode, this is an old x-ray of what this looks like. Yeah, you got crazy ass teeth, look at that. So it would stimulate and activate different parts of the brain. And they found that if they placed the electrode in areas known as the septal region and the medial forebrain bundle, that rats would want to self-stimulate over and over and over again. So we're going to look at a little bit of what that looks like. So this is showing the location of where this is found. So they place this electrode, connected the electrode to the lever, and this is the graph. This is showing it over about an hour and a half time, and they turned on and off the bar. So this is like an internal control to see what the rat would do if there's no actual activity coming out from touching the bar. So when the bar was on was during these empty spaces, and when the bar was turned off, where it was not stimulating the brain, was in these hatch spaces. The rat didn't know whether it was doing it one way or the other, except to find out that, like, when he touched it, it was giving him a sense of reward. And you can see that in the spaces where the bar was actually doing something and stimulating his reward systems, the number of bar presses, so this is cumulative bar presses, so it just goes up and up and up as the rat does more and more bar presses. And you can see when it's on, the rat is pushing that bar over and over and over and over again. You have to look at the scale here. 500 bar presses, 1,000 bar presses. The rat reaches about 1,000 bar presses in about 45 minutes. That's insane. So what is that per minute? That's about 2.5 times a minute. No, 20 times a minute. Yeah, like every 3 seconds. Totally crazy. Boink, 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 boink. Yeah. That rat really liked to push that bar. But then as soon as it turned off, then the rat is no longer pushing the bar. You can see it's like basically flat. You can see the rat tries a couple of times, like, okay, I want some of that juice. And turn it on briefly, then it's like, okay, showtime. And then turn it back off, and then the rat stops. So the rat is in love with self-stimulation. So the areas that are being stimulated in the brains of these rats are ultimately the dopaminergic areas of the brain. So we've talked extensively about the substantia nigra pars compacta and its contributions to regulating motor learning. We haven't talked about in detail at all the contributions it makes to reward-related cognition, not in much detail, but it does play an important role in that as well. So its reward component to just other cognitive features, including habit-related features around drugs and drug addiction. It plays a role in reward outside of just motor learning. But we're not going to really discuss that much. Instead, we're going to focus today on brain circuits related to the ventral tegmental area. So this is another dopaminergic area of the brain, very close to the substantia nigra pars compacta, and it regulates reward-related cognition, including wanting and liking, and positive reinforcement. So this is where it's located. And let's remove the basal ganglia related parts. Instead, we're going to focus just on the ventral tegmental area and related areas. So the ventral tegmental area is this reward-related area. And the medial forebrain bundle is the thing that was being stimulated by this the electrode in those rats. It's a group of descending myelinated fibers innervating the VTA. And it's perhaps the most effective site of stimulation. So this stimulation activates the VTA directly. So when you stimulate the forebrain bundle here in the septal region, so all these descending inputs into the VTA, and now the VTA is going to start firing. The VTA receives excitatory input from a number of brain areas, including the prefrontal cortex, and it receives inhibitory input from the nucleus accumbens. And so we're going to talk about connections between nucleus accumbens, the ventral tegmental area. We are also going to talk about how the VTA has projections to two major pathways. There's the mesolimbic pathway and the mesocortical pathway. So the mesocortical pathway, importantly, the mesolimbic pathway is VTA to the nucleus accumbens, and the mesocortical pathway is VTA to the prefrontal and frontal cortex. It makes extensions all the way down into other areas of cortex as well, but especially into the prefrontal and frontal cortex. There's huge dopaminergic input into those areas, and that comes from the VTA. So we're going to talk about the activity that happens in the VTA over the process of learning and how the VTA responds to reward and lack of reward. So in this experiment, we're recording in the VTA of a monkey. And in this experiment, so we're going to look at the activity that happens before there's any learning that's actually occurred. So there's no prediction of a reward here. The light isn't being turned on yet. It would be, so this is before teaching the monkey that the light predicts the reward. So we're just looking at what happens when the monkey gets a reward. So the juice dispenser moves forward, the monkey gets a little bit of juice, and as soon as he gets that juice, there's increased activity in the VTA. You can see this is the histogram here. So this would be the accumulation of all these raster plots here. Remember, each line is one individual trial. And they did it looks like 15 times or something like that. So, and then you can see that activity is increased pretty soon after the reward is given. Probably on the order of tens of milliseconds. So that's before there's any sort of association, before the monkey has learned that the light gives you a reward. If you teach the monkey that the light predicts the reward, something interesting happens to the VTA. So the monkey has made the association that the light, when it gets turned on, it's going to get a reward a few seconds later. And now the VTA fires when the light turns on. So as soon as the light turns on, so that's what this line means, now the VTA increases activity with the onset of the light. And interestingly, it doesn't increase activity with the reward. It has now sort of shifted its emphasis to the light, since the light is going to give us this is the first thing that tells us, hey, we're going to get a reward. Something even more interesting happens in the next part of this experiment. So in this experiment, now we're going to turn on the light, but we're not going to give the monkey a reward. So we're flipping the script on this poor monkey here. So the light turns on, and there's an increase in activity here, right? Because the monkey is expecting to see some kind of reward, but it doesn't get a reward. And instead, now when it doesn't get a reward, the activity in the VTA actually goes down. So there's a decrease in VTA activity, which means it's decreasing the amount of dopamine that it's giving to these reward circuits. So it's actually like a sign of disappointment because it expected a reward, but it didn't actually get it. And so now this is one way of sort of breaking apart that association that had been made into that monkey's brain, that the light will give us a reward, and now when we take it away, well, okay, maybe the rules have been changed. So the VTA, it works as a predictor. It can work as a predictor of reward, and then it also can tell us when the reward is not there. So the next part, and a very important part of this, the mesolimbic pathway, is the nucleus accumbens. So the nucleus accumbens is part of the striatum, and it is filled with GABAergic medium spiny neurons. So it receives input from the VTA, just as the substantia nigra pars compacta projects to medium spiny neurons in the dorsal striatum. So it's very, very similar, okay? So this is known as the nigostriatal pathway. This is the mesolimbic pathway. So this pathway from the VTA connecting to the nucleus accumbens. This is the mesolimbic pathway. This regulates liking and wanting. It's especially important in making memories during classical conditioning. So it is these areas that are going to be very active during that classical conditioning phase. The medium spiny neurons and the nucleus accumbens have D1 or D2 receptors, and D1 is associated with a reward, and D2 is associated with aversion. Very, very similar to what we saw when we talked about the basal ganglia. That D1 reinforces circuits and D2 breaks down circuits. It works in a similar fashion in the mesolimbic pathway. So this is a diagram, a more simplified diagram that I have made to show the various connectivity that happens within this pathway. Okay, so the VTA, it responds to novel stimuli and unexpected rewards, and has reduced activity when expected rewards are not present, right? So that's what the VTA does. So it has increased activity to novel stimuli. It likes new stuff, getting juice especially. But when you take away novel expected rewards, it decreases its activity. So that means increased activity of VTA increases dopamine release into the nucleus accumbens and in the prefrontal cortex. So these are dopamine neurons sending a bunch of dopamine into the nucleus accumbens. There are also connections from the VTA to the prefrontal cortex. So it also gets a bunch of dopamine up into the prefrontal cortex. And this dopamine release gives a feeling of satisfaction and reinforces behavior and makes associations. So the nucleus accumbens receives input from a bunch of different brain areas. The hippocampus, the amygdala, the prefrontal cortex, and the thalamus. They all provide input, just sort of like how there are cortical areas that provide input into the striatum in the basal ganglia. Similar system happening here. And so it's going to be driving input to these medium spiny neurons, and these medium spiny neurons can then operate to influence the activity of the ventral tegmental area. We will be looking at a slide a little bit later that will show that the synapses and connectivity between the nucleus accumbens and the VTA, it isn't exactly like the striatal substantia nigra pars compacta connectivity. There's room for opiates and other different kinds of neurotransmitters in this circuit. So it's a little bit more complex. What I want you to know from this is just a little bit of this connectivity of the mesolimbic pathway, these connections from the VTA to the nucleus accumbens, that the nucleus accumbens has D1 and D2 medium spiny neurons, but there are also these connections from the VTA up to the prefrontal cortex, and this is the mesocortical pathway up to the prefrontal cortex. And that there's a bunch of cortical and major glutamatergic inputs to the nucleus accumbens itself. So I've talked about microdialysis already. I mentioned it briefly when we talked about the dopamine areas in the song control system, right? That we can measure dopamine locally. So I'm going to walk through this briefly again, that we could measure. So in this experiment, what's happening here is that we're measuring the amount of dopamine in the nucleus accumbens. We know that it gets input from the ventral tegmental area, but we're going to prove that activity in the ventral tegmental area will increase amount of dopamine in the nucleus accumbens. We can also just look at changes in dopamine in the nucleus accumbens when animals go through different conditions. And so we're going to look at some of the data related to that. So, and the way microdialysis works is that you have this probe that goes in, and it's a very, very thin probe, put it into a particular brain area, and there's going to be a diffusion of certain kinds of ions that go through there, and they will be swept up into the cannula. So you have this like flow happening here with some things coming out, and then with the absorbance of compounds like dopamine being taken up, and so you have this exchange that happens here, and so there's an input that comes in, and then there's output that comes out, and you can measure the output over time. So you keep like track of the little drops that are coming out, and you can see changes in dopamine over time. So this is showing some data from this, And this is showing in this kind of experiment where you give an electrode into the the medial septal area. And so this is that bundle that would be synapsing onto the ventral tegmental area. And then we look at dopamine release in the nucleus accumbens, right? Because the ventral tegmental area projects to the nucleus accumbens. What you can see is that during phases of self-reinforcement, when the reinforcing brain stimulation is turned on, dopamine levels shoot up 400%. And then when the reinforcement brain stimulation is turned off, it declines. And it declines over some time. So the rush of getting all of that dopamine lasts for a little bit, but the euphoria declines pretty quickly. And then you can see it can go back up again. So self-stimulation of VTA leads to dopamine release in target regions like the nucleus accumbens. So this is showing exactly what we were looking at before with self-reinforcement, except with different amounts of current. So we can change the amount of current that gets applied to the VTA, and you can see that in this case, with increasing amounts of current, we get increasing amounts of dopamine released, and that it declines over time. And then this is just showing the number of stimulations that these rats, I think these were rats, that they were doing per 15 minutes. So in this case, with the highest stimulation for the for the septal region, you can see that these rats were doing nearly 1600 stimulations per 15 minutes. So nearly 100 stimulations a minute. That's insane. That's a lot. That's a lot of stimulation. This is showing that this effect is dose dependent. That more stimulations equals greater dopamine released. So because there's more current being released, it feels better. So it has this positive reinforcing loop where the rat is now pounding that lever, getting that rush over and over and over and over and over again. And this leads to a huge release in dopamine. And over, you can see just a very brief period of time, and then it declines relatively quickly. There are various lines of evidence that support the idea that dopamine mediates reward in the brain. So self-stimulation is facilitated by giving drugs that enhance dopamine. So you can improve self-stimulation by giving animals amphetamine, MAOIs, dopamine transport blockers. So would mean that the dopamine is present for longer. So this would be like dopamine reuptake inhibitors. So drugs that deplete dopamine, such as rezapine, inhibit self-stimulation. So if you give something that breaks down the dopamine, now they're not so interested in doing self-stimulation. And when you treat animals with dopamine antagonists, this also blocks the reinforcing effects of self-stimulation. So, for example, applying a dopamine receptor antagonist in the nucleus accumbens blocks the effects of medial forebrain bundle self-stimulation. And so infusion of a dopamine receptor blocker here means, and so the brain has been turned around here. This is the VTA. Here's the nucleus accumbens. When you infuse a blocker right here, now they're not so interested in doing reinforcing the brain stimulation. Natural rewards will increase the amount of dopamine present. And so we're now looking at the amount of dopamine being released in the prefrontal cortex, as well as in the nucleus accumbens, in the presence of food. You can see that there's an increase in both the prefrontal cortex when they eat. So there's a big increase. So F is when they're feeding. You can see that there's it goes up to 200%, basically doubling. Then it goes back down to normal basal levels after about 2 hours. Feed again, goes up again to 200%. Natural reward, food is delicious. Same is true, although to a slightly lesser extent in the nucleus accumbens. Sex also will increase dopamine. So in this figure, so we can just look at the control initially. You can see that the amount of dopamine present in when there's a female present, when there's copulation, you can see that there's an increase in the amount of dopamine present. And during the mounting and intromission, it then eventually decreases back to baseline. So during the copulation phase, the dopamine is highest and it's relatively high. During the intermission, and eventually goes down. In the presence of amphetamines, the amount of dopamine released is even higher. And these animals will have more mounts and intromissions during that time as well as more ejaculations. Good times. So neurotransmitters that are involved in the reward pathways are shown here in this table. Dopamine, clearly, very important. It acts on receptors D1 and D2, and its function is pleasure, euphoria, mood, motor function. Serotonin is also important to this. We'll briefly talk about it. And we're going to talk a lot more about serotonin when we talk about mental disorders. It has receptors, one of which is serotonin 3. There are others. And it seems to be related to mood and impulsivity, anxiety, sleep, and cognition. And then there's also endogenous cannabinoids. The endocannabinoids is also what they're known as. They act on receptors CB1 and CB2, and they play a role in pain, appetite, and memory, but they also have euphoric effects as well. And then there's also the opioid peptides. We've talked about these a little bit. There's endorphins, enkephalins, dynorphins. They act on receptors, opioid receptors, kappa, mu, and delta. In general, they're related to pain, but they also play a role in modulating the circuits within the reward pathway. So, but in all rewards, dopamine is the final activation chemical, but it's not the only one that's involved. It's not just dopamine. So now we're going to talk about drugs and how it affects these reward circuits. Here's a nice list showing a list of common drugs. We've got cocaine and amphetamines that they act on dopamine. They also can affect GABA, and that happens in the amygdala. I'll let you look through this list. We got the opiates, you got nicotine, the cannabinoids, and alcohol. And we'll talk about these in a little bit more detail in a circuit. I believe it's in the next slide. Yeah, this is it. So this is where I wanted to walk through some of the actions of addictive substances on the brain. Despite a diverse initial actions, addictive substances produce some common effects in the VTA and the nucleus accumbens. Namely, they increase dopamine transmission. So that's ultimately the goal of most of these stimulants. So or drugs, I should say. So stimulants like cocaine and amphetamine, they directly increase dopamine transition in the nucleus accumbens. So the stimulants, they can act on the axon terminals to force dopamine out. That's how amphetamines work. So they get into these dopaminergic neurons from the VTA, and they release and force out dopamine out of the ends, and so there's a lot of dopamine here at the nucleus accumbens, acting on dopamine receptors. Cocaine acts as a dopamine reuptake inhibitor, and that means that dopamine is now sitting at the synapse a lot more than it should. Opioid peptides act both in the VTA and the nucleus accumbens. So we got opioid actions here on neurons that synthesize opioid peptides within the VTA. They will release opioids that will induce changes within these neurons on GABAergic neurons. And you can see that the opioids will act on these circuits. So opioids, alcohol, and inhalants, such as the solvent toluene, they do the same indirectly. Alcohol also activates the release of opioid peptides. So it acts on these opioid generating neurons, releasing opioids into these circuits, ultimately leading to an increase in dopamine in the nucleus accumbens. So heroin and prescribed opioid pain relievers, they directly activate opioid peptide receptors, so they act just on these receptors themselves, whereas alcohol will induce the release of endogenous opioids. Nicotine acts on dopamine neurons within the VTA. So here's the nicotinic receptors. This is part of the acetylcholine pathway. So there's acetylcholine that can act on neurons as a modulator, and this is the nicotinic version of the cholinergic receptors. Nicotine acts on those receptors, inducing dopamine, exciting these neurons to release more dopamine into the nucleus accumbens. Now, cannabinoids are a little bit more complicated. They may act in the VTA to activate dopamine neurons, but they also act on nucleus accumbens neurons themselves as well. So there's cannabinoid receptors in both on the GABAergic neurons that are interneurons in here, and they likely serve to shut down GABAergic signaling here so that the dopamine neurons are a little bit more excited, so they release more dopamine. They also have effects on the nucleus accumbens neurons themselves. So in summary, alcohol, what it does, it actually serves to inhibit GABAergic neurons. We learned about that when we talked about the cerebellum. It also does this within this reward circuit, and so it inhibits those GABAergic neurons that project to the dopaminergic neurons in the VTA, and so now there's more dopamine released. Heroin binds to the opioid receptors that inhibit GABAergic neurons that project to the dopaminergic neurons in the VTA. Cocaine blocks the function of the dopamine transporter, so this is the protein that would take up dopamine. And then nicotine activates the cholinergic neurons that project to the dopaminergic neurons of the VTA. Dopamine-enhancing drugs, they enhance self-stimulation behavior. So in a self-stimulation experiment, rats were permitted to apply microstimulation to themselves at will. The rate at which they applied microstimulation depended upon the stimulation frequency. So first, we're going to talk about cocaine. So that's in the first experiment, and let's just walk through it a little bit. So in this case, we're looking at the responses per minute in the presence of cocaine or a baseline. And this is the stimulation frequency down here. You can see if an animal was on cocaine, that it required lower stimulation frequencies in order to induce a greater response. So stimulation frequency, so like 30 hertz or something would give you almost maximal response from the animal, nearly getting 60 responses per minute while the animal's on cocaine. And this would not be sufficient to do anything to an animal that has nothing. But eventually you can get the frequency up high enough where now the animal is now doing self-stimulation itself. So cocaine can change that. Nicotine does something similar. So you can see self-stimulation here. At the starting point for the baseline, it's around, 35 hertz or something like that. You can see that's nearly at already maximum in the nicotine animals, and there's hardly any self-stimulation happening in the controls. But eventually you can increase the amount of activity getting into the nucleus accumbens, and eventually both animals, whether they're on nicotine or not, will now do self-stimulation. But the whole take-home point of this is that when animals are on these drugs of abuse, that it lowers the propensity for animals to do self-stimulation-like behavior. So rats can be trained to self-administer virtually all addictive drugs, including opiates, amphetamines, cocaine, alcohol, and nicotine. They usually need repeated exposures, and addictions are then marked by escalation in dose habituation, tolerance, and then finally, resistance to extinction, when behaviors are no longer deliver drugs, but deliver punishment and foot shock instead. So they will, um, instead of getting any drug, if you start to deliver a punishment, they still will try to get the drug because now they're addicted, even though it's just leading to them getting a foot shock. So that's resistance to extinction. So let's talk about the different stages in addiction. First, there's explorative, where drug taking is found to be pleasurable. Then there's positive reinforcement, which encourages repeated drug taking. Eventually, you develop habits. Unfortunately, that also leads to tolerance. So the more you take a drug, you're going to change the receptor expression within the brain, and then you need more and more of the drug to get the same kind of high. And eventually, this leads to withdrawal symptoms, which causes stress. And by stress, I mean like the kind of sympathetic nervous system stress symptoms. So let's talk about a little bit of what drugs of abuse do to dopamine. If you remember the levels that we talked about with food and sex, increasing amount of dopamine in the brain around 150% to up to 200%. You can see that in the accumbens, also in the caudate, so there's changes that happen within the basal ganglia as well. Time after amphetamine, after 1 hour, 2 hours, 3 hours, 4 hours, 5 hours. And you can see that animals that get amphetamine have a huge increase in the amount of dopamine being released in the nucleus accumbens, 1,000% higher. Cocaine, not quite as high as amphetamine, but still incredibly high. I know I shouldn't say that. So between 1 and 2 hours with cocaine, you can see it's up to past 300%. Whereas, with food, we saw the most that it got was around 200% or 150%. In the nucleus accumbens, it was like 120%, right? So we see similar effects with other drugs of abuse as well, such as nicotine. So just nicotine alone, increasing the amount of dopamine released in the nucleus accumbens and in the caudate, up to 200% immediately. And then here's ethanol. It's kind of cut off down here at the bottom. You can see. So ethanol also increasing the amount of dopamine being released. And we can see that there's a dose-dependent response. So this is the highest concentration here, next highest, and then the lowest concentration shown here. You can see that it's about what you would see with feeding at a relatively low concentration with enough alcohol. Now we're talking about real dopamine release. So one way of seeing how a circuit has changed is to look at the AMPA to NMDA ratio. As you learned in Neuro 1, when you have increased activity at a glutamatergic synapse, a lot of glutamate being released, it binds to an NMDA receptor. If there's enough, and if this happens enough times, you'll get an influx of calcium into the postsynaptic site, which will induce the activation of kinases, which leads to the recruitment of AMPA receptors to the postsynaptic site. So now you've had more AMPA receptors, so you're going to get more glutamate signaling going through to the postsynaptic cell. So what happens in the presence of drugs or, exposure of drugs? So in this case, I'm going to walk through some results from one paper showing some really cool results for this. What they did is they treated mice with drugs. They just gave them drugs once. 24 hours later, they killed them. They made brain slices containing the VTA. And then they recorded dopaminergic VTA neurons. And they looked at the AMPA to the NMDA ratio. They wanted to see how excitable are these dopaminergic VTA neurons. So normally, you can look at the current of a cell in isolation. That's what the dual is. That means you're looking at NMDA and AMPA current. Then what you can do is you can put on an AMPA blocker. And so you're just looking at NMDA, and NMDA is always bigger. So you can see that the NMDA current is quite big. And then you put on an NMDA current blocker, and then you remove the AMPA blocker, and then you can see the AMPA current. And you can see in general, and usually in the presence of like an animal that was injected with saline, the AMPA to NMDA ratio is something like 0.4. AMPA is about 40% the height of NMDA. What does this look like in drugs? So let's take a look. In the presence of cocaine or amphetamine, so these are animals that got one dose of cocaine and amphetamine, and then they looked at the AMPA to NMDA ratios later. You can see now that the AMPA ratio is much bigger. So there's been a lot of activity happening within the circuit, and you can see that the synapses are now changed. And here's the quantification for this. You can see cocaine and amphetamine, the AMPA ratios are now around 0.7, so around 70% of the current for NMDA, whereas normal is around 0.4. So there's been a big change in these VTA neurons. We can look at other drugs of abuse. So here's the control, saline, presence of morphine, so it's an opiate. You can see 0.7. Nicotine, 0.7. Ethanol, 0.7. All these drugs of abuse are increasing the AMPA to NMDA ratio, suggesting that these connections are strengthened, which will mean that these neurons are going to be more excitable and release more dopamine. So other mood-altering drugs, such as fluoxetine and carbamazepine, which is an anticonvulsant, sodium channel blocker, I believe it's also used in treatment of schizophrenia. You can see that there's no increase in the AMPA to NMDA ratios. So these are mood-altering drugs, but they don't have any euphoric feeling. There's no sense of reward by taking fluoxetine, which is just Prozac. You can see no real change. And you can see the AMPA to NMDA ratio is exactly like it is in the controls. So this shows that these circuits can be changed in the presence of drugs of abuse, including just once. Doing it once will change your brain. So we can also look at the behavioral context for this as well. We can do a place preference test with rodents. So you have a 2-chamber cage, which will have different colors and textures, light conditions, other stimuli that will allow rats to distinguish one side from the other. They can say, this is the checkered side with a grid floor. Here's the light, there are lights in this room and no checkered and it's not grid. So these are distinctions that'll just let the rat know, like, oh, something is here or here. Rats will prefer to spend time in places where they had pleasurable experiences. So if you give a rat a bunch of delicious food in this side, he's going to want to stay in this side and not spend so much time in this side, because he's hoping that, hey, you know what, maybe I'll get some more delicious food if I hang out in here. So what happens with drugs? Let's take a look. So rats that had a dopamine agonist infused into the nucleus accumbens prefer to spend more time in the compartment where they received the agonist before. So in the drug-impaired compartment, they're spending more time. You can see just total amount of time, and it's not even, in this case, not even a real dose-dependent response. I mean, it does look like it increases from here to here, but this is about the same. But compared to controls, nothing. You can see it's a significant increase relative to controls. So in the next several slides, we're going to talk about what happens with self-reinforcement when drugs of abuse are now leading to addiction and leading to dependence. And we've got a cycle that happens here, and there isn't any like one real start to it. It's a series of things that just sort of cycle in when addiction has set in place. So we can, just kind of start, we're going to talk about binge and intoxication. So this is the stage at which an individual consumes intoxicating substances and experiences rewarding or pleasurable effects. So the basal ganglia and the reward centers are critical for this. Then we have withdrawal and negative effects. So you take something pleasurable and then it goes away. This is the stage at which an individual experiences a negative emotional state. in the absence of the substance because they're sad that they're not high. And then this leads to preoccupation and anticipation, the stage at which one seeks substances again after a period of abstinence. So we have this, and then that will lead to more binging and intoxication. And you do that, eventually it goes away, then you have withdrawal, you get negative affect, and that leads to preoccupation and anticipation. And there are different brain areas that are involved with this process. The progression of drug dependence over time has this shift that occurs. There's this declining shift in overall mood, and it changes over time. So from initial use of drugs, there's a positive reinforcement that happens. You have a lot of pleasurable effects. So the very first time that you're using drugs, mood is actually much higher than normal, your homeostatic point. Okay, so that's like homeostatic point here. And so that's the first wave of using drugs. But eventually you undergo withdrawal, and now your mood is actually less than homeostasis. And this is going to lead then to relapse and use of the drugs. But now you need more of the drug during this time because you have started to develop some tolerance, and you're never going to get quite that same high that you did the very first time. Eventually, this wears off, and then there'll be some relapse that occurs, and you can see that the mood is just going down lower and lower and lower. We have increasing dynorphin, norepinephrine, CRF, and decreasing dopamine and GABA over time. What you can see is that with a substance use disorder, the initial phase, you have a lot of positive reinforcement. This is going to increase our motivation to use drugs because it feels good. And so the intensity increases as well. But eventually, tolerance starts to set in, and we start to have withdrawal effects. And eventually, we were leading not so much to be driven by positive reinforcement, but by trying to reduce negative reinforcement. So this negative mood, you can see by the end, at late dependence, when the brain is significantly changed, when the person is high, the high is not even close to a normal mood. But this is as good as the person's going to feel, because when withdrawal hits, it's actually even worse, and it's well below the range of overall mood. And so these are negative effects, and an individual will want to avoid those negative effects, so they will use drugs to avoid negative reinforcement. And so you get this transition that occurs when drugs are no longer very effective, keeping a positive reinforcement, and instead we just get negative reinforcement. There are individual differences in propensity for addiction in rodents. There's also differences in humans as well. So in a study, Daly et al., published in Science, they tested rats for impulsiveness. They found some are high impulsive and others are low impulsive. So they had this test, and they found that high impulsive rats have fewer D2 receptors in the ventral striatum. So remember, D2 receptors will work as the inhibitors of reinforcement. D1 would be kind of the positive rewarder, and D2 would be the negative. And so this was this is the high impulsives here. This was the low impulsives. You can see that these are brighter, and so that was more D2 versus these guys. And interestingly, these guys also have they escalate cocaine use. So the black is the high impulsive, and number of cocaine infusions per hour. So both groups are getting access to cocaine. You can see that across different sessions over time, that the high impulsive groups tend to use more cocaine during each session because they are impulsive. We know that in humans, of course, there are individual differences as well. Some of this is related to overall temperament, overall social development, our relationship with drugs and alcohol very early on. We know that those are factors. Comorbidity is an important thing. Anxiety disorder, personality disorders, mood disorders, they can contribute to drug use and misuse. Genetics is another factor to this. We don't know all the details of it, but it appears that about 40% of total variability is associated to genetics as far as drug dependence is concerned. And then there's also certain protective factors too. So it can work on the other side as well. Some individuals are less prone to being addicted, and this is also contributions from genetics, personality, and environment. So over time of addiction, there are major changes that can occur in the brain and overall brain structure. There's evidence that prolonged drug use results in both structural and functional brain changes. One example of this is that a decrease in Krebs transcription factor within the nucleus accumbens, as well as in the amygdala. There's also decreases in metabolism with the orbitofrontal cortex. So within drug addiction, overall metabolism is decreased. The orbitofrontal cortex, as we're going to learn in the next lecture, is really important for decision making and logic. A very important part of the prefrontal cortex. And there's also decreases that happen in D2 receptor binding. So these are fMRI images of that compare the brain of an individual with a history of cocaine use disorder. And the brighter the signal, like the more red it is, the more D2 receptors that are located there. And you can see that an individual who is using cocaine for just 1 month has fewer D2 receptors, so less of the aversive control, so D2 receptors, remember, they serve to act to inhibit certain changes. And by this decrease in D2 receptors, now you're losing some of that aversiveness within the system. And this is 1 month after cocaine use. So this is an individual who's been abstaining from cocaine for 1 month. And then also the same individual imaged 4 months later, and you can see that their D2 receptors started to increase a little bit, but it's still substantially reduced compared to a control subject. You can see that they have not returned to normal baseline levels, even after 4 months of abstinence. And so this can lead to very long-term changes in brain structure, and this is one reason why you can imagine people undergoing relapse over time. So one of the last phases is withdrawal. So you got these changes that happen in the brain. Of course, then there's also the withdrawal phase. So what's happening with this decreased mood? Well, one part of this is that corticotropin releasing hormone is actually activated. So this is an activation of the overall stress pathway, the HPA axis. So this is a figure I took that shows CRF. It's known as CRH. So this whole system will activate the sympathetic nervous system. It'll also lead to the increase in expression of ACTH, leading to increased expression of corticosteroids. It also so activation of the amygdala, which will influence the how our mood and overall feelings and anger is being regulated. And then CRF also acts on to brain areas to lead to sympathetic system activation. So during these phases of withdrawal, you're basically having a severe stress response with increased heart rate, increased blood pressure, increased blood glucose, and an increased response to stressors. So if your overall baseline level is super stressed when you're undergoing withdrawal, you're going to just be very sensitive to stressors as well. So this is, the major changes that are occurring here is essentially you're just activating the stress response during phases of withdrawal. So like these down phases right here, 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 getting progressively worse and worse. Each time, even more worse than the previous. So this stage of the addiction cycle is characterized by the disruption of executive function caused by a compromised prefrontal cortex. So the prefrontal cortex is affected here. We'll talk about the role of executive function in the next lecture and the role of the prefrontal cortex in that. But this is going to be leading to then preoccupation and anticipation. Activity of the neurotransmitter glutamate is increased, which drives substance use habits associated with craving. It disrupts how dopamine influences the frontal cortex. So even normal contributions through the mesocortical pathway from the VTA to the prefrontal cortex is severely disrupted. Then you have overactivation of decision-making circuits in the prefrontal cortex promotes habit-like substance seeking because you become preoccupied with drug seeking. And then underactivation of the circuits in the prefrontal cortex to regulate mood and emotion. And this leads to the promotion of impulsive and compulsive substance seeking. So you got overactivation of decision-making circuits that related to habit, and underactivation of circuits related to regulation of mood and emotion. And these are the kinds of changes that you associate with severe drug addiction. So that's the end of this lecture. What are the definitions of a reward? How do scientists identify the reward areas of the brain? What are the major areas of the mesolimbic and mesocortical reward pathways? How does the response of the VTA change with respect to presence, absence of a reward, and if the reward is paired to a stimulus? What are the major inputs and outputs from the nucleus accumbens to the VTA? What are the neurotransmitters? What are the effects of natural rewards on dopamine release? What are the effects of drugs on dopamine release, and where do they act? Specifically looking at slide 21. What are some of the changes that happen in the brain post-chronic drug use? What are some of the factors that make some individuals more susceptible to drug addiction? And what are some of the changes associated with withdrawal? So that's it. Yep. Don't do drugs.